Hey everyone, this is our presentation. It's called DRUGsophila, and that'll make more sense as we get further along. So without further ado, here we go. What's up, guys? I'm Marco Gambino. I was born and raised in Brooklyn, New York, which happens to be my only personality trait. I'm a sophomore studying neuroscience, and I have a double minor in biology and history. And in the future, I want to do something either concerning neurodegenerative disorder research with either Alzheimer's or Parkinson's disease. And I read an interesting book over the summer called The Performance Cortex, which discussed this interesting intersect between sports and neuroscience. So that's something I would also like to pursue.
Hi, my name is Adana Thomas. I am from Beaufort, Georgia. I am currently a sophomore in neuroscience with a minor in linguistics. And my future career aspirations is that I want to do neurodegenerative disorder research, as well as something with linguistics, like neurolinguistics.
Hello, my name is Serena, and I'm from Columbus, Ohio. I'm currently a sophomore studying neuroscience on the pre-med track, and my clinical aspiration is to become a pediatric neurologist.
Hi, my name is Ziyi, and people always call me Emily. I'm from China, and I'm a sophomore. I major in neuroscience and psychology, and currently I'm on the pre-med track, but I'm also interested in pharmacy, and my research area would focus on neurodegenerative disorder and emotional disorder.
So our project mainly involves Parkinson's disease research, which, if you guys don't know, Parkinson's is a neurodegenerative disorder concerning dopaminergic cell loss in the substantia nigra and the striatum. And symptoms of the disease include bradykinesia, which is slow movement, resting tremor, and there's also some non-motor dysfunctions that are less important to our study. And as of right now, there is no cure, but there's a lot of research being done looking for both a cure as well as treatments for the disease.
The animal model that we will be using in lab is the Drosophila melanogaster. It is a fruit fly model that's very popular in lab settings because they're fairly low maintenance and they have a short lifespan, around 8 to 14 days, as well as they're highly fertile. Females will lay about 30 to 50 eggs daily, so it yields a lot of test subjects in a short amount of time. They also share a 75% similarity for genetic sequencing of diseases with humans, which makes them a good model to study human disease research in lab. They're also very easy to cross. Mutations are very straightforward when crossing these models. We are going to be using in lab the GAL4 UAS system.
We will be using the GAL4 UAS system to create our crosses for our project. GAL4 is a gene normally found in yeast and is inserted into a fly genome where a native enhancer will control its expression or inexpression. UAS, also known as upstream activation sequence, is a section of DNA that, when bound to by GAL4, promotes the transcription of a desired reporter gene. By using the GAL4 UAS system, the crosses that we are creating in our experiment should allow us to produce flies with Parkinson's disease and also allow us to visualize the healthy and unhealthy dopaminergic system.
We crossed the GAL4 PAM with each of the UAS line. The anatomical system we targeted on is the dopamine transporter PAM neurons, which is short for DTPN due to its association with to learning, memory, and locomotive functions. The dopaminergic PAM neurons are part of the adult dopaminergic mushroom body input neurons, which is shown on the image on the right. Mushroom body is a center for associative learning that has been implicated in a wide range of fly behaviors, including olfactory learning, context generalization, habituation, temperature preference, and sleep. On the left, it is a cluster of approximately 100 cell bodies in the cortex of anterior inferior medial protocerebrum of adult brain. There are a lot of types, and almost all of them target on the medial lobe.
So the first way that we're going to manipulate the PAM system is using UAS tetanus toxin. So basically for our flies, we want to infer Parkinson's disease. And in order to do that, we have to shut off the dopamine system. And that's what this does. So tetanus toxin, it chronically silences neurons. And it does this by cleaving a snare protein, which is involved in vesicles binding at the synapse. And without the snare protein, the vesicle can't bind. So therefore, no neurotransmitter can release into the synaptic cleft. And that's how tetanus toxin shuts off the neurons. And that's how, if the cross works out, the flies shouldn't be able to generate enough neurotransmitter, or any neurotransmitter in that case, thus shutting off the PAM system and inferring Parkinson's disease.
Another cross that we're going to be looking into is the UAS EGFP cross. EGFP stands for Enhanced Green Fluorescent Protein. It's one of the two proteins involved in bioluminescence in a certain type of jellyfish named there. It's used to image living tissue using fluorescence microscopy. It's a very light-sensitive process using a fluorescent microscope. What's pictured here is our own dissected and imaged EGFP brain.
For one of our crosses, we will be using the UAS YFP tagged light-activated halorrhodopsin chloride channel. Halorrhodopsin is a ligand-gated ion channel that operates on the concept of optogenetics. By using optogenetics, this allows us to manipulate specific genes to transiently express or inexpress behaviors by shining a light. In our case, we will be using this to inactivate the dopamine system in response to shining a red light onto the flies.
In our experiment, we tested the impact of caffeine and alcohol on Drosophila behaviors, and the Drosophilas are dopamine deficient. Alcohol has both positive and negative reinforcing actions in the brain. Caffeine is not a depressant, it's a central nervous system stimulant. It is the most consumed psychoactive drug. It's used to promote wakefulness, enhance alertness, and reinforcement effects. Like other stimulants, caffeine enhances dopamine signaling in the brain.
All of our crosses involve a Gal4 dopamine PAM female. And then the first cross we're going to make, we're going to cross that with a UAS tetanus toxin, and that's going to infer Parkinson's disease in the flies. The second cross is an EGFP cross, which will allow us to visualize a healthy system using fluorescence microscopy. And the third cross involves a YFP tagged light-activated halorrhodopsin chloride channel, which will allow us to visual an unhealthy system after shining red light onto the fly brains to turn off the dopamine PAM neurons. And then the yellow fluorescent protein will allow us to view that under fluorescence microscopy.
Because diseases are sequenced so similarly between Drosophila and humans, this makes them a viable model for our Parkinson's disorder research. And we are looking into what types of drugs that affect the dopaminergic system will help treat Parkinson's disorders. For this lab, for our own purposes, we are going to be using a stimulant and a depressant.
The two main types of flies that we will be working with are wild-type and Parkinson's-induced flies. Both of these flies will be observed under three conditions: sober, caffeinated, and intoxicated. We have 6 different fly groups that we will be observing. Our first group of flies is sober wild-type, which will serve as our baseline group. And our other test groups will be our control and Parkinson's disease flies with the conditions mentioned earlier. For our fly preparation procedure, we will be socially isolating and/or starving the flies depending on the assays that will be performed. On the image on the right, you can see an example of how we isolated the flies in preparation for the behavior assays.
Basically, we used our phones to record the videos, and the videos were uploaded to Google Drive for later analysis. And all the data obtained is all based on observations from the videos, and we use references for analysis.
So the first assay that we did was a learning and memory assay, and we chose to do this because dopamine actually plays an important role in learning and memory. So since we shut off the dopamine systems, we figured why not see what happens. So the setup was we put a 3D printed maze inside a petri dish with agarose, and then we covered it with the other half of the petri dish so the fly couldn't escape. And what you see in the center of the maze is actually a cup with food, apple juice, and for the male trials, a decapitated female in the cup, so that the flies would be attracted to it. And for extra motivation, we starved the flies overnight so that they would actually be hungry, so they would want to complete the maze. And so for recording the trials, we started by aspirating the fly. Then we started a stopwatch, and when they reached the middle of the maze, we would stop the timer and record the time and we would kind of average out the times to see if their learning had increased and if their memory had decreased as a result of the Parkinson's disease and also the drugs that they were manipulated with. And unfortunately, flies are really, really bad at mazes. Like, they just kept wandering on the walls, going absolutely nowhere. So there were no completions, and therefore, unfortunately, there was no data that was usable for this assay.
Another behavior that we looked to observe in our experiment was the hunger behavior of the Drosophila. So Parkinson's disease was observed to decrease appetite in patients, and alcohol and caffeine are appetite suppressants. So we wanted to observe the changes in the behaviors of the hunger behaviors of the Drosophila if we were to give them alcohol and caffeine. We wanted to see if disinhibition would occur or if no changes would be made. Our setup is a fight chamber, which are 3 microscope slides superglued into the formation of a triangle halfway filled with agar, and a vial of apple juice placed in the center. We will starve the flies before each of the trials, and we recorded 3 trials with 3 different fight chambers as pictured in the slide, and we recorded that for 15 minutes. Our results were that the Parkinsonian flies didn't eat anything, and the wild type flies, when given caffeine or alcohol, also did not eat, but the sober wild type flies did eat. I interpret that as a success because it means that our crosses worked. The Parkinsonian flies did not eat, and caffeine and alcohol do seem to suppress the appetites of the flies, as the caffeinated and drunk flies did not eat.
Another one of our behavior essays was used to study aggression. We will be observing the aggressive behaviors of the flies by placing them in a fight chamber similar to those used for the hunger assay. We will also be placing a food vial in the middle of the chamber with a headless female on top to encourage the male flies to fight over, as shown in the figure on the top right. Prior to the essay, we will be starving and socially isolating the male flies. For the essay trials, we will be recording 20-minute videos using smartphones, which will be analyzed and scored. We will also be running 3 trials for each of our test groups. For our results, we expected that the wild-type intoxicated flies will exhibit less frequent aggressive behaviors compared to our baseline group, which is supported by the data shown. This is interpreted as ethanol playing a huge part in impairing locomotive functions in high concentrations. The flies exhibited slow and lethargic behavior, resulting in less aggressive interactions. We did not have enough time to run our caffeine wild-type group. However, we do predict that there will be an increase of aggressive behavior due to caffeine being a stimulant. Unfortunately, we also did not have enough time to run our Parkinson's disease flies. However, based off of previous research relating to this topic, and based off the data we have collected so far, we expect to see the pattern of aggressive behaviors of our wild-type flies to be reflected in our PD flies.
We conducted a courtship assay to examine how alcohol affects courtship behaviors of dopamine-deficient flies, because the courtship behaviors depend on the level of dopamine. It is affected, but not like all determined by the dopamine level. Courtship behaviors, including chasing, also orienting, tapping, singing, and you can see their wings are vibrating and leaking by extending proboscis, and finally attempting copulation, and we'll see mountain behaviors. And the mean duration time is about 20 minutes, and the maximum can reach 40 minutes. For the first trial, it's just a wild-type flies. The fly chamber will be placed in the light to minimize the time for the male to find the target female. Each isolated naive male and female was collected from their own tube, and they were transported to the flight chamber, which is the same size as the aggression assay. And the behaviors were recorded for 20 minutes. For the second trial, the wild-type flies were transported to the chamber with alcohol dissipated in the air for about 5 minutes, and we follow the same steps in trial 1. And also the behaviors were recorded for 20 minutes. We calculate the courtship level through the CL index, which is a measure of the fraction of time the male fly performed the defined behavior over the fixed observation period, which is 20 minutes. We predict the result to be that both female and male with dopamine neuron loss are expected to show diminished courtship behaviors. For the results, I can't draw any conclusion from the video I took because even if they are just passing by, they don't have any obvious interaction. And where we can see from here, they're just passing by and not showing any behaviors at all. So I analyzed the possible reasons why the trial wasn't successful because previous researchers have shown that no enthusiasm is shown until 2 days after the pupae has hatched. The time for pupa to hatch takes about 8 to 14 days, so we don't really know what day they're hatched. Like, time varies a lot, and they could be just hatched on the day we use them. So, because we collected those pupa 4 days before the experiment, and we don't really know. We basically can make sure they're 2 days after they're hatched. So, if we have enough time, we could wait longer. And second reason would be we could have design a better chamber for them because the chamber we used is the same size as the aggression assay, which would be a little bit large for the courtship assay. It takes more time for those two flies to meet, and we could have used a cap which is about a 5 milliliter diameter, and I believe if we use a smaller size, it should minimize the time for them to interact.
So after gathering all the data from the assays and performing all the tests, unfortunately, the results were not as expected. The drugs really didn't help so much as we would have liked them to, but that's also because caffeine and alcohol weren't the drugs that we originally wanted to do research on. It was actually marijuana, because as of right now, there's lots of research going on using medical marijuana in Parkinson's disease, and there are videos on the internet that show after patients will smoke some marijuana, their tremor will die down, and it seems like a very promising field. So I figured that if we were to continue this research in the future, we would actually either manipulate the cannabinoid receptors in the flies' brains or just give them marijuana extract to see how repeating the experiments using marijuana as the drug of choice would impact their Parkinson's disease and their symptoms as well as their behaviors.
Thank you for listening to our presentation. As a closing, I found some fruit fly memes off of the internet that I thought I would enjoy. It's a cute little infographic here. Little fly getting crunk, and a fly drinking coffee, fly getting injected, fly drinking coffee, caffeine right there. All ties back into the experiment. These two memes are just about how short their lifespans are. And then this one in the middle I really like because it's a pun. It's fruit in an airplane, fruit flies, right?